Alright, here we are in Chapter 9, Nutrition and the Human Digestive System. And we are in Theme 2, the second chapter of Theme 2, which is Physiology of Humans and Animals. Okay, so here you see these a lot of subtopics, but actually this chapter is a very interesting chapter because all of us like to eat. Right, let's look at 9.1 digestive system. We are going to look at the structure of the digestive system. So this system is made up of a long, muscular alimentary canal that starts from the mouth and ends with the anus. So the mouth is the inlet and the anus is the outlet. So the parts that form it includes the mouth, we have the esophagus, the small intestine, the large intestine, and also the anus. So the other organs in the digestive systems include the liver, we have the gallbladder, and also the pancreas. So another one is that we have the glands, salivary glands, we have the gastric gland in the stomach, and also the intestinal glands in the small intestines. These glands are important because they are going to secrete digestive juices into the alimentary canal. Okay, so another picture of the digestive system. Okay, next let's look at the types of digestion. Okay, so first we have to define what is digestion. Okay, you have to mention that it is actually a process, okay, that breaks down large and complex pieces of food into smaller and simple pieces that can be dissolved for easy absorption, and you know that it has to be absorbed into our bloodstream. Digestion is made up of two parts, okay, namely the physical and also the chemical digestion. So what is physical digestion? It is the mechanical breakdown of food to form particles by means of chewing, okay, using our teeth, and also the peristalsis action. Okay, chemical digestion involves chemical, and that is actually referring to the enzymes. And it is actually the decomposition process of complex molecules into the simple ones. Okay, let's start with the digestions of carbohydrates in the mouth. Okay, digestion actually begins in the mouth and not in the stomach. So the presence of food in the mouth stimulates the secretion of saliva from salivary glands. Okay, when you're about to eat, when you see the food, that moment you feel that your mouth will be watery, isn't it? Now this statement explains that. Okay, and we know saliva contains amylase, salivary amylase, so it is very useful. Okay, this is actually the enzyme that is going to hydrolyze starch into maltose. Okay, hydrolysis, that means this starch is being broken down, lysed, okay, in the presence of water, okay, and becoming maltose. Do you still remember maltose? It is actually one of the disaccharide. If you have forgotten about it, maybe you will need to go back to chapter 4. Okay, the pH of saliva ranges between 6.5 to 7.5, which is about neutral, and that is actually suitable for this enzyme to act. Okay, it is the optimum enzyme for this. It is the optimum pH for this enzyme to react. Okay, digestion of carbohydrates in the mouth. So just now, saliva also helps to form bolus, helps the food to form bolus. So that means ball in the form of ball to make it easier to be swallowed. Okay, so when swallowing happen, what at the same time, what will happen is that the epiglottis will close the trachea. Okay, I'm sure you remember what is trachea. So why is it closing the trachea? It's because it wants to prevent the food from entering into the wrong pathway, okay, from entering into the trachea. Okay, otherwise, if it enters into the trachea, what happens is aspiration. So what is aspiration? Okay, aspiration is when food, drink, and other foreign objects goes into the lung because they went to the wrong tube just now. Okay, so this is why epiglottis will close the trachea whenever you swallowed. So in the esophagus, the food bolus is moved by peristalsis. So this food bolus moves by means of peristalsis. So you still remember what is peristalsis? It is actually the rhythmic contraction and relaxation of muscles along the alimentary canal. Okay, and for your information, peristalsis not only happen in the esophagus, but it actually happens throughout the alimentary canal. Okay, so you will see here muscular walls of the esophagus contract over this area, and at the same time, the muscular walls of the esophagus relax at this region. So here it goes down along the way, okay, until it reaches the next compartment, which is the stomach. So it is rhythmically contracting and relaxing. All right. Okay, so here's a brainstorm question. Okay, you chew slowly a small piece of bread and you observe the taste when you first tasted chewing and after a few minutes of chewing. So is there any difference when you start chewing and then after a few minutes of chewing? Okay, so do you actually feel that it tastes that it is actually getting sweeter after some time? Okay, this is because salivary gland secretes an enzyme known as salivary amylase, and this amylase is going to break down starch into simple sugar, okay, which is maltose. It is the disaccharide. Okay, and if you have forgotten the characteristics of the disaccharides, again you will need to go back to chapter 4. Okay, one of the characteristics of the disaccharides is that it tastes sweet. Okay, and this explains why after some time of chewing it tastes even sweeter. All right. Okay, the digestion of protein in the stomach. Okay, so moving on, let's look at the stomach. So the stomach is actually has a thick wall and also sausage shaped organ. And these walls contain gastric gland which secrete gastric juice. So the surface of the stomach wall is lined with epithelial cells, okay, that have undergone adaptations in the structure and also function, forming the glands. Okay, so you can see here over here, this is the walls of the stomach. You can see the tiny boxes over here. These boxes, this lining is actually the epithelial cells, which has undergone adaptation in structure and also function. So there are three types of cells being shown here. The first one, the chief cells over here, is the one that secrete pepsinogen. It's a type of enzyme, but is inactive. Okay, and next, this is parietal cells, which is the one that is responsible to secrete hydrochloric acid. Okay, and also we have the mucous cells that will secrete mucus. Okay, here we are. This is the stomach. This is the esophagus just now entering into the stomach, and this is the duodenum, which is the first part of the small intestine. Okay, so pepsinogen, do you still remember? Just now we mentioned about it. It is secreted by which cell? Pepsinogen. Okay, go back. This is the one, the chief cells. Okay, the chief cells is the one secreting pepsinogen, but then it is inactive. It has to be activated by hydrochloric acid to become pepsin. There only pepsin can function to hydrolyze protein, pepsin, hydrolyze protein to smaller polypeptide chains. And this hydrochloric acid, you still remember that it is secreted by these parietal cells. Okay, let's look at hydrochloric acids. Actually, our stomach has got this hydrochloric acid, which is very acidic. Okay, the first function, it has to prepare a medium with a suitable pH for the pepsin to act. Okay, so pepsin has to act in pH that is very acidic, 1.5 to 2. Okay, another function of hydrochloric acid is that it has to stop the enzymatic action of the salivary amylase just now. Okay, from the mouth, which is alkali, so it has to stop this action. Okay, and the third one is that it will kill the bacteria because it is very acidic. Right, so sometimes when you realize that you eat those foods that are not so clean, but then you don't get food poisoning, everything is fine. Maybe because when the food reaches the stomach, all those bacteria are being killed by the very acidic acid which is in the stomach. Okay, so all right, activate enzymes. Just now we mentioned about the pepsinogen, isn't it? Pepsinogen will have to be activated to pepsin before it is able to just now to break down the protein to become the polypeptides. So here is it. This in the presence of HCl hydrochloric acid. So this is now become active. This is inactive form. This is active. In the old syllabus, we also mentioned about the renin, okay, which is also present in the gastric juice. But now in the new syllabus in KSSM, renin is not mentioned. Okay, alright, yeah, this is from the old syllabus where these two enzymes are said to be present in the gastric juice. Okay, so maybe you can read as some extra information for you. Okay, so this is still the same. In the old syllabus, it says it will hydrolyze the large protein molecules to become the smaller polypeptide chains. So this remains unchanged lah. Okay, next is on the function of mucus. Okay, so remember mucus is secreted by which cell, which epithelial cell? Yes, by the mucus cell. So the function is to protect the stomach wall from the reaction of the hydrochloric acid and also the digestive enzymes. You imagine if our stomach is to be opened up and all the enzymes and also the hydrochloric acid spill out. It is able to corrode the surrounding, because it is very acidic. But then, surprisingly, our stomach is okay. It is fine. Okay, why? It is not corroded. The wall is not corroded is because of the presence of this mucus. You see how important is this mucus? So the food in the stomach is mixed with gastric juice made up with hydrochloric acid and also pepsin. Right? In the stomach, this is what happened. Then food is churned by peristaltic action of the stomach wall for a few hours. Okay, and then the contents of the stomach will finally change to semi-fluid, and we call it as chyme. So slowly it will enter the duodenum, which is the first part of the small intestine when the sphincter muscle relax. Okay, so this is sphincter muscle acting as a valve to control the flow of the partially digested chyme from the stomach to the small intestine. Okay, so here to highlight this title is supposed to be here lah. Function of the mucus. So the first lines answer the function of the mucus. Okay, next, this is what you can see, churning of the stomach. So after you feel after eating, you see this stomach is actually not static, but then it is moving, and not to say move from left to right, but then the movement is called churning. Okay, to form a semi-fluid known as chyme. So the stomach muscles contract, the pyloric sphincter relax and secretes the chyme into the duodenum. So when these muscles over here relax, this chyme will move on to the next compartment, which is the small intestine. Okay, so this is how it goes. All right, so next, just now we start with the mouth, and then we have the stomach. After that, we have the Yes, now we are entering into the small intestines. So we'll be talking about also not only carbohydrates, proteins, but also lipids. So lipids starts here, like the digestion starts in the small intestines. So the small intestine has got these three parts: duodenum, jejunum, and ileum. And duodenum is the first part. So it will receive the semi-fluid food from the stomach, just now the chyme. Also, it will receive the bile, which is produced by the liver. Also, the pancreatic juice, which is secreted by the pancreas. So, duodenum is receiving all these different chemicals from other places to carry out its function. Okay, so this is what will happen in the duodenum. We have these three um, enzymes: LAT lipase, pancreatic amylase, but this A is because of the amylase over here, and trypsin. So you know this trypsin, right? You have heard of this. This is to digest. This is involved in the protein digestion. Okay, just now we have salivary amylase. Okay, you still remember salivary amylase function, which is to hydrolyze starch into maltose. So same here for pancreatic amylase, also the function is also to hydrolyze starch into maltose, but then it further hydrolyzes it lah. So that means maybe the one in the mouth is not properly hydrolyzed. And then when it goes to this duodenum, um, this hydrolysis of starch takes place also. Okay, so LAT. This is the one in your textbook. It says components that are involved in the digestion that takes place in the small intestine, the duodenum. Okay, so the chyme over here just now it receives the bile from which is stored in the gallbladder, but the bile is produced by the liver. Okay, also it receives these juices, pancreatic juices from the pancreas. All right, so pancreas secretes pancreatic amylase, trypsin, and lipase into the duodenum through the pancreatic duct. So the LAT just now. So in the liver has a lot of function actually, not only the one which is stated here, a lot of functions. Maybe you can read on that for further knowledge. Okay, extra knowledge. Okay, so liver here, it produces bile. It's not the gallbladder. Gallbladder is the place where it is stored. So liver produces bile stored in the gallbladder, and then this bile, when it is in use, it will go to the duodenum. Okay, so it flows into the duodenum through the bile duct. What is the function of bile? It's actually to neutralize the acidic chyme. Okay, neutralize the acidic chyme, which is from the stomach just now. So it also prepares an alkali condition for the enzyme action in the duodenum. Okay, alkali condition, because just now in the stomach it is very acidic. So it has to prepare an alkali condition for the enzyme to function in the duodenum. Also, bile will emulsify lipids by breaking down lipids into tiny droplets, and this will increase the surface area for the lipase activity. Okay, so this is what happened in the duodenum. We have the LAT, lipase, amylase, and also trypsin. So pancreatic amylase is what I've mentioned just now, hydrolyzed starch into maltose. Trypsin, hydrolyzed polypeptides into shorter peptides. Because just now initially protein digestion has started in the stomach. We have the pepsin, remember, that hydrolyzed protein into polypeptide. So from there just now the polypeptide here will then be further hydrolyzed by trypsin to peptides, even shorter than polypeptides. And also we have the lipase, which will hydrolyze lipids into fatty acids and glycerol. So the digestions of lipids starts in the duodenum, which is the small intestine. Okay, so this is the one. Okay, information which I've mentioned just now, you can read on. Okay, now the glands on ileum wall secretes mucus and intestinal juice. Intestinal juice that contains maltase, sucrase, lactase, lipase, and erepsin. Maltase, sucrase, lactase, erepsin. 1, 2, 3, 4. Another one is lipase. Okay, because from the old syllabus, last time when they mention intestinal juices, only these 4. But then in KSSM, we notice that there's another additional of lipase over here. Okay, so not only these 4, but also the lipase is included in the intestinal juices. So how many juices now? We have the gastric juices, we have the pancreatic juice, and also we have the intestinal juices. Okay, so intestinal juice, let's look at erepsin. By the name itself, just now it will further hydrolyze peptides into the smallest subunits of the protein, which is amino acids. And so to say that the digestion of protein will be completed here in the ileum. Okay, still remember ileum, that is the third part of the small intestine. We have the duodenum, jejunum, and also ileum. Okay, maltase, by the name itself, it will hydrolyze maltose, okay, into glucose. Glucose is already the simplest form, and so to say it is completed here at ileum. Okay, we have sucrase, also will hydrolyze sucrose, okay, to become glucose and fructose. Sucrose is a disaccharide. Okay, lactose is also a disaccharide. So it is hydrolyzed by lactase to form 2 monosaccharides, glucose and also galactose. So we have all these end products. The smallest or the simplest form is already here in the ileum. And so to say, we understand that absorption will happen here in the lowest part of the small intestine, and that is the ileum. Okay, because digestion has already been completed at this region. All right, and in ileum also alkaline medium is needed for the enzymes to act optimally. Okay, carbohydrate digestion. Now this is the one which is similar to the equation over this slide. Okay, this is from the textbook. Erepsin. Yeah, erepsin. Yes, okay. So, yeah, next is on absorption.